Okay, so the Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. So topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support early career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. So please see the website for more details. So I'm Dr. Irundika Dias. I'm from Aston University based in UK, based in Birmingham in UK. And I'm also part of the Biochemical Society Research Panel Area 6. So in this webinar, we will hear from Dr. Benjamin Brzezinski-Falcon, who will discuss his group use of cryo-EM to determine the structures of pathological protein assemblies. So Ben received his PhD in molecular biology from the University of Cambridge and subsequently carried out postdoc at the LMB, where he helped determine the cryo-EM structures of assembled tau in neurodegenerative disease. So since October 2019, Benjamin had led a research group at the LMB with a focus on the molecular mechanisms of protein assembly in neurodegenerative diseases. For his research, Ben has been awarded several prestigious awards, including the Alzheimer's Research UK Rising Star Award and SCORE Young European Researcher Prize. He was also elected to the EMBO Young Investigator Program in 2022. Please, questions will be asked after Ben's presentation, but you can send your questions during the talk. If you have a question, please type it in the question box as shown in the image in the screen, and we will try to answer as many times as the time allows. So with this, now I'm going to hand over to Ben.
Okay.
Yeah, everything looks
Thank
fine.
you very much. Hopefully you can see my screen and you can hear me. And it's a pleasure to be here today. So yes, I'm going to talk about the atomic pathology of neurodegenerative diseases and the way that we've been getting new insights into this by doing cryo-EM of amyloid filaments. And hopefully this title will make sense as I go through my talk. So if I just get the pointer. So I'd like to sort of start here and make the point that really the hallmark of all common neurodegenerative diseases is the abnormal assembly of proteins. And there are not very many of them that undergo this abnormal assembly. They're shown here in bold. So we have things like tau and SOD1, TDP43. And they assemble and form inclusions either within neurons and glial cells, as is the case with tau, or in the extracellular space of the brain, as in the case of prion deposits and those of amyloid beta. And the first point I'd like to make here is that these proteins have very little in common in health, in their physiological function. So they carry out a broad variety of roles that are not related. So it's really the abnormal assembly that ties these proteins together in disease. The second point is that there are mutations within each of the genes that encodes these proteins that give rise to autosomal dominantly inherited forms of various neurodegenerative diseases. And this really tells us that protein assembly is fundamental to neurodegenerative disease and must play some sort of causal role, although we don't fully understand what that is. I'd also like to point out, and you'll see this from the list of the different diseases next to each protein, that there is assembly of different proteins that can converge into one clinical presentation of a disease. For example, TDP43, FUS and SOD1 in ALS. And then conversely, there are multiple diseases that can result from the assembly of only a single protein, such as tau, alpha-synuclein, and TDP43. And this is somewhat surprising that one protein can go on and give so many different pathologies in the brain. So we want to understand in the lab and in the institute the molecular mechanisms of neurodegenerative disease. And to me, that really means, for the reasons I've just laid out, we need to understand the molecular mechanisms of protein assembly. Now at the LMB, many of the groups, and our group included, take a structural biology approach to these problems of molecular mechanisms. And this idea, applying this to neurodegenerative disease and to protein assembly, is really not a new idea. In the LMB, this was started by Aaron Klug in the mid-80s, and then really worked on throughout the 30 years or so up until today, especially driven forward by the work of Tony Crowther at the LMB. But it really wasn't until about 2015 when the possibility of getting proper full structural resolution of these protein assemblies became a reality. And that was really enabled by the so-called revolution in cryo-EM, which you may have heard of. So there were big steps made in both the hardware, the direct electron detectors, for example, in cryo-EM, and in the software to reconstruct 3D protein structures from these cryo-EM data. And at about that time, there are also big strides made by Chowdhury and Shaw-Skeres's group in applying these reconstruction approaches to helical assemblies as well, which is especially relevant for these assemblies in neurodegenerative disease. So in around 2015, in a collaboration between the groups of Michel Goda and Shaw-Skeres, and involving myself as a postdoc in Michel Goda's group, and another postdoc, Anthony Fitzpatrick, in Shaw's group, we attempted then to solve the structure of assembled tau protein in Alzheimer's disease. And this schematic or workflow here shows you how we went about doing that. So we would start with fresh frozen post-mortem brain tissue from an individual who had died of Alzheimer's disease. So if you like, we're using the primary isolates, the primary material, and the native assembly structures. This turns out to be very important, as we'll see later on. We would extract them using a mild solubilization with a detergent and then differential centrifugation. I'll go into this a bit more later on. We could then, using these advances in the cryo-EM hardware, collect data sets, and then using this helical reconstruction in the program Rely on, determine 3D structures of these filaments. And that looks like this. This is a paired helical filament of tau in Alzheimer's disease. You can see two C-shaped subunits or protofilaments. You can see that shape perpendicular to the helical axis. So this was seen already by Tony Crowther in the LMB. But what we could now do is build in the atomic model of tau within these C-shaped protofilaments. You can see the residues of tau that form this filament fold, and we can see the secondary structure of these assemblies. And that is principally what we call an amyloid filament. And what we mean by that is that these folds are enriched in beta strands, and then as you stack identical folds from different tau molecules on top of one another, you end up with these intermolecular beta sheets running along the filament axis. And that's the hallmark of amyloid filaments. There's a separation then between the molecules by about 4.7 angstroms, just the separation of strands within a sheet. Now, what we see for tau and many other amyloids, although this is not universal, is that if you see here in the cross-section of the filament, the main chain can fold back on itself, and then you get the lateral stacking of beta sheets perpendicular to this helical axis, and you get a 10 angstrom component to this amyloid filament structure, which is the separation of beta sheets dictated by the average separation of side chains of residues within these sheets. Now, since showing that this was possible and doing this for tau, Michel and Shaw's group have gone on to really look at tau assemblies in all of the tauopathies almost, and they're listed here. Many acronyms which you don't need to worry about, but these are all different diseases defined by tau assembly. Here we have Alzheimer's disease with that C-shaped protofilament fold that I showed in the previous slide. And they've also done this for alpha-synuclein. So I was involved in the tau work. And what we found is that for each disease, we have a different filament fold. And you can see that in these schematic representations here. And so these are almost the same residues of tau forming these different folds, although we do see different isoforms contributing to different folds in different diseases. But you have similar residues, and yet they're able to form many different folds, which initially was a surprise. And then even more surprising is that for every individual with a given disease, they will have a certain fold, and that differs between diseases. Now, this is unexpected, and it suggests that they have some importance in disease. And of course, what we'd like to understand is how do these folds form in the first place, and how do they influence diseases? Now, just one hypothesis is that these could be related to prion-like strains, whereby the fold itself will somehow impart on the disease progression and the formation of different diseases. But of course, to really test these hypotheses that come out of the structures, we're going to need some sort of experimental models that reproduce these very many different filament folds. We can't rely on human brain samples at the end stage of disease to go forward with this work. And so this is just from a recent review that I wrote with Michelle and Shores, where we identified this as a grand challenge in the field of neurodegenerative disease research, where we would like to have models either purely in vitro with purified proteins through to cell and animal models that can reproduce these structures that we see in different diseases. We can use cryo-EM then to check whether these models are faithfully reproducing these folds. And when we find those that do, we can then subject them to a whole barrage of molecular biology and cell biology investigation to understand, is there formation? How do they form? Do they spread in different ways? And is their toxicity different depending on the fold? So that's very much a future work that we are involved in. Now, another sort of immediate consequence of having these structures is that we can use them to try to find binders to these assemblies that could either be used for diagnosis. So here on the left-hand side, I have an example of PET ligands that can bind to these filaments and can be used to diagnose individuals. And then on the right-hand side, the other kind of binder would be compounds that can disassemble or prevent the assembly of these proteins as a therapeutic strategy. So I just highlight two of the early papers using the tau paired helical filament structure I showed at the beginning to do this. And the way in which these studies went forward is to then solve additional cryo-EM structures with these lead compounds bound. And that reveals, as shown here, where these compounds are binding, and that can then be used to improve and design additional generations of these tools. Now, the other approach that is starting to be used as well is to use the structures then in silico screens for binders, taking advantage of the atomic coordinates that we have generated from the cryo-EM. And we'll see this coming through alpha-synuclein, and as we'll talk about later, other proteins like TDP-43 as well. Now, I'm going to sort of step back, if you like, and talk about the work that has come out of my lab since we started this back in October 2019. And having come with a background in tau, I switched focus to the protein TDP-43, the tRDNA binding protein 43. So back in 2006, two studies, one by Manuela Neumann from the group of Virginia Lee and another from the group of Masato Hasegawa, showed that the assembly of this protein TDP-43 was the hallmark of almost all cases of the motor neuron disease and myotrophic lateral sclerosis. This is probably the most common neurodegenerative disease of midlife, and about half of cases of a neurodegenerative condition called frontotemporal lobar degeneration, which is the cause of frontotemporal dementias. And so I showed you in the tau slide, we had many of those diseases I showed you were types of frontotemporal lobar degeneration with tau assembly, but actually half of frontotemporal lobar degenerations have TDP-43 assembly as their core pathology. There's also a more recently described entity called limbic predominant age-related TDP-43 encephalopathy, and this is a clinical mimic of Alzheimer's disease that appears to be driven by TDP-43 assembly rather than tau and amyloid beta assembly, although this is still very much an active area of investigation. But if true, this would make this by far the most common TDP-43 proteinopathy. We also find TDP43 assembly as a co-pathology in many diseases that I've referred to as mixed diseases down here. Well over half of cases of Alzheimer's disease show TDP43 assembly, for example, and it's been shown that where TDP43 assembly is present, this accentuates the disease and leads to worse disease outcomes. Now, the other reason that we became interested in TDP43 is because the evidence on its structure of the assembled form in disease was quite contradictory and uncertain, and it looked like things may be quite different to tau and alpha-synuclein, which piqued our interest. So what did we know about TDP43 assembly prior to our studies? Well, we knew that it was abnormally post-translationally modified. So here's a domain organization of TDP43. You can see it's an RNA binding protein with 2 RNA recognition motifs shown in green in the center here. It has this Dix domain at the N-terminus. So this is a ubiquitin-like fold that enables head-to-tail oligomerization in health. So it's not a monomeric protein. It has then this low complexity domain. So a low complexity domain is just a region that has a distribution of amino acids that's skewed towards certain amino acids that we wouldn't expect. So not all low complexity domains are the same. And in the case of TDP43, we have an overrepresentation of glycines, of hydrophobic residues, and of glutamine and asparagine residues. So the first abnormal post-translational modification, and the most striking, is truncation from the N-terminus. You can see that removes the Dix domain, the nuclear localization signal, and the first RNA recognition motif, and then parts or all of the second RNA recognition motif, leaving behind a shared fragment of the low complexity domain. We also see abnormal phosphorylation at the extreme C-terminus at 6 sites that are shown here. So these have been confirmed to be specific to disease, although there probably are others that remain to be identified. And in addition to these modifications, there is ubiquitination and possible acetylation that's disease-specific as well. Now, not long after the work from Virginia Lee and Masato Hasegawa's labs had identified TDP43 and these modifications, many other studies looked at the ultrastructure of these assemblies using thin section fixed EM on human tissue sections. So here's a postmortem human tissue, brain tissue, and you can see here one TDP43 inclusion on the left, and at high magnification on the right, you can see this is indeed a mesh of filaments. They decorate with gold conjugated TDP43 antibodies. So TDP43 assembly in disease is forming filaments, but there were 2 sort of negative, confusing pieces of evidence out there in the field. The first was that these filaments bind these so-called amyloidophilic dyes very, very poorly, if at all. So we're talking about things like Congo red and thioflavin T here, and their derivatives, like the PET ligand that I showed you for tau in the previous slide. And there's one study here that was comparing binding of thioflavin S, for example. And so this led many to conclude that actually, although filamentous, these assemblies were probably not amyloid, and therefore we'd be thinking of something quite distinct from tau and alpha-synuclein. And the second piece of evidence, or lack of evidence, I suppose, was the inability to isolate these pathological assemblies at the expense of the physiological form of TDP43. So I've mentioned that TDP43 has this Dix domain. It also has this low complexity domain, both of which will multimerize to some extent in health. So this is forming somewhat insoluble complexes in health. So the kind of protocols that we would use for tau filaments, for example, to extract out based on the density or size of these aggregates was really not efficient for TDP43, and therefore a lot of studies would mix the physiological and pathological form of this protein, making it very difficult to interpret what really is disease-related. So that was the first challenge in the lab, was to find a extraction method where we could get the pathological form of assembled TDP43 and leave behind the physiological form. And so we teamed up with Masato Hasegawa, who of course had been involved in the first discovery of TDP43 and had really done the best, I think, biochemical work on TDP43 assembly in disease. So he had a protocol that was working quite well for extracting this, and we then joined him to optimize this to be used with cryo-EM. And essentially, it is very similar to the method used for alpha-synuclein and TDP-43. So again, of course, starting with human brain material, because we don't know how well these experimental models will recapitulate the disease. And it's using a solubilization in the detergent sarcosol plus differential centrifugation. But we really had to optimize those steps, when to add the detergent, at what concentration, and how to carry out the differential centrifugation. And we then checked on the success of these attempts by following this on Western blots. You can see in the total homogenate that's predominated by the physiological form of TDP-43 at 43 kilodaltons, plus you can sort of see a bit of the C-terminal fragments here. And then eventually we were able to leave that physiological 43 kilodalton form behind in a soluble fraction, and to bring in all of the assembled TDP-43 into this final, what we call insoluble fraction. And you can see that the phosphorylated TDP-43 in this plot using a phosphorylation-dependent antibody will then only detect this insoluble fraction. So when we then put this on an EM grid, either by negative stain or cryo-EM, we could suddenly see those TDP-43 filaments that we knew existed from the earlier thin section negative stain EM studies that had been carried out in 2008. And indeed, we can do immuno-EM on these filament extracts and label them with gold conjugated TDP-43 antibodies, as you can see here on the right. And this then enabled us to make cryo-EM grids where we could find the filaments, and we can then apply this pipeline for determining these filament structures as we did for tau and alpha-synuclein. Now, I should point out that the abundance of the filaments, or lack of abundance of TDP-43 filaments in the brain, is quite striking compared to tau, probably 2 orders of magnitude lower concentration of TDP-43 filaments. That's reflected in the thin section EM, where the inclusions are much less densely packed. And so that really made this challenging from a cryo-EM processing point of view. And it was a postdoc, Diana Arseni, the first postdoc to join the lab, who took on this challenge and had to collect hundreds of thousands of micrographs and manually annotate the filaments within these micrographs in order to determine the structure. So a real feat of processing of cryo-EM data here. So that paid off for Diana, and she was able then to crack the structure of TDP-43 filaments. She had focused on cases of ALS that also had frontotemporal lobe degeneration of a type B that we refer to as type B. I'll explain this a bit later, but essentially about 50% or more of ALS cases will have this FTLD, and we think of this really as a spectrum of a single disease, ALS and type B FTLD, rather than 2 separate disease entities. Now you can see at the far left that we have these helical filaments. You can see TDP-43 molecules, if you squint, stacked up along this filament. So this does look like an amyloid filament, and indeed, if we look at the cross section here and we look at the schematic on the right-hand side, you can see that the TDP-43 is forming many beta-stranded regions, and as these stack different molecules on top of one another, you'll get those intermolecular beta sheets that define these filaments as being amyloid. Despite that, they are lacking this 10 angstrom component of amyloid, so we don't see beta sheets coming together perpendicular to the helical axis. And that's really because of this low sequence complexity of the sequence that forms these. So it is the first half of the low complexity domain that forms these filaments. We could see that from the structure. That makes a lot of sense because it can incorporate all forms of TDP-43, all species, the full length and the C-terminal fragments, all of which are present in disease. And it can incorporate the abnormal phosphorylations at the C-terminus as well. So the ordered course stops just before we find phosphorylation. Indeed, just showing here on the right-hand side, you can see this really is a low complexity sequence. So about a quarter of the residues are glycines, a third neutral polar, mainly gluteins and asparagines, and about a third hydrophobic. We have a couple of prolines and a single charged residue, this arginine here, exposed to the solvent. And so this kind of explains why we don't have stacking of beta sheets. There's glycines that break up beta strands and introduce many kinks and turns within the structure. And it may also show why amyloidophilic compounds don't bind to these filaments. And that's because we think because the surfaces of these filaments are incredibly smooth compared to those of other filaments like tau and alpha-synuclein. You can see many glycines, which of course lack a side chain, so that's the smooth surface. And then these neutral polar residues, glutamines and asparagines, they're able to form hydrogen bonds with the main chain and flatten themselves off. I'm just pointing here at the bottom here of this fold, you can see one of these residues that's doing that, one of these asparagines that's doing just that, forming a hydrogen bond back into the main chain. So in the previous slide with the PET ligands bound to tau filaments, you could see that these ligands intercalated in between grooves in the filament side chain formed by large charged amino acids, and they simply don't exist in TDP-43 filaments. So that's our hypothesis and something we're following up at the moment as we try and find binders to these filaments. Now, Diana didn't just look at one sample of brain. She instead, for a single individual, looked at two regions, the prefrontal cortex, which is of course associated with the frontotemporal lobe degeneration, and the motor cortex associated with the ALS. And in both of these regions, she found an identical fold. And I'm just showing you now, this is still the cross section, but the raw data of the 3D reconstruction. So hopefully you can make out that double spiral fold. And then Diana also looked at a second individual, and here she obtained the prefrontal cortex, and again found an identical fold. So what we think we have is an identical filament fold or structure shared among individuals and brain regions, those who have ALS and FTLD type B. Now, as I mentioned earlier, I'll explain a bit more about FTLD with TDP-43. And we talked about the type B form, but there are actually multiple types of frontotemporal lobe degeneration with TDP-43 assembly. And these were put together in a consensus report by Ian McKenzie in this paper here. And he designated them as type A, B, C, or D, and there may be a type E as well. So these are principally defined based on the location and distribution of the assembled TDP-43. Firstly, at the cellular or even subcellular level, where the TDP-43 can form either cytoplasmic, nuclear, or neuritic inclusions, and you can see they're differentially present in these different types. And then at the tissue distribution level, where especially type A, you have a concentration of the assembly within layer 2 of the neocortex. That's also seen to a lesser extent for type C, whereas type B and D have a wide distribution throughout the cortical layers. In addition to this, there's distinct genetic variation that associates with these different types, principally mutations in progranulin, which are always associated with type A, mutations in VCP, which are always associated with type D, and then the expansion of C9orf72, which can either be associated with type A or B. Type C is distinct in that there is no known genetic variation. And finally, suggesting that these are different diseases rather than only different neurodegenerative disorders, they are also associated with different clinical presentations of frontotemporal dementia and ALS. And as I mentioned, type B is unique in that it is associated with ALS, whereas the other types are associated with different forms of frontotemporal dementia. And for type D, it's associated with what we call a multi-system proteinopathy, where it can present in many ways, including as a myopathy. So having determined the structure of TDP-43 in ALS and type B FTLD, we were interested as to whether it would form a similar structure in these other diseases. And we were thinking about tau and alpha-synuclein at the time, where we knew that there were different filament folds of these proteins in different diseases. But also, subsequent to publishing work on type B and ALS, others had come out to suggest that actually maybe amyloid filaments were unique to this type, and we'd find something quite different in type A FTLD, for example. So we focused on this as the most common type of FTLD with TDP-43 pathology. And again, Diana used this protocol that we developed to extract the filaments and determine their structure using cryo-EM. And this is what she found. So she looked initially at 2 individuals who had progranulin mutations and 1 individual that had sporadic disease. So what we mean by that is there was no known genetic variation associated with neurodegenerative disease. And then subsequent to this publication, she's also looked at 2 individuals with C9orf72 expansions who also have type A FTLD TDP. And I hope you can appreciate in all cases we have an identical filament fold, and this is regardless of the genetic variation present in these individuals. So this is then in detail what that fold looks like. You can see again it's the first half of the low complexity domain. It's slightly longer this time, so it extends about 10 amino acids towards the N-terminus. And you can see again it's an amyloid filament because it has these much beta content, many beta strands. You can see actually now we have the stacking of beta sheets. You can see beta strands running anti-parallel to one another within this fold, and actually many regions of that. So they're very, very different to the filament fold in ALS. In addition to this, we saw additional densities shown in light yellow here on the left-hand side. Some of these are non-proteinaceous molecules, which will also have been seen in many other amyloid filament structures and may be important to their formation, but which we cannot identify by cryo-EM. They're too small and they don't follow the helical symmetry necessarily of these filaments, and that will require additional techniques to identify these, but they may be important. So just to compare then these 2 folds, we can see, of course, visually in these schematics, the folds are very, very different on the global level. And this is reflected by different secondary structure. So the secondary structure is either turn regions or beta strands. And you can see in the sequence alignment at the bottom that actually they don't align, almost don't align at all between the 2 folds. So regions that form strands in one fold will form turns in another. There's really a very high level of structural plasticity, if you will. Something unique to the type A fold, we found local alternative conformations, and they're shown in these grayed out regions in these 2 parts of the fold. And so this is unexpected. It shows actually amyloid filaments don't have to be stacks of identically structured molecules. You can have local variation. And the structures actually revealed that probably post-translational modifications dictate this variation. And just to highlight that at the N-terminus here, we had an arginine in one of the conformations that's completely buried without charge compensation. And we found that this is citrullonated, so it's lost its charge, and that enables this conformation. Whereas arginine in the second and this alternative conformation, we think is monomethylated. We also found evidence for that. That's too bulky to fit into this conformation, but it's fine here in this alternative conformation because it's solvent exposed. So we have post-translational modifications that are leading to changes in structure and may be important for the formation of these structures. And of course, we now need to investigate that in models that reproduce these filament folds. Now, as I mentioned earlier on for tau and alpha-synuclein, it's very much the same case for TDP-43 that the structures that had been solved of in vitro assembled TDP-43 filaments were completely different to those that we found in the human brain. You can just see that here, showing you the various different structures. This one in the bottom left is actually a solution NMR structure of soluble TDP-43. And just to show there's an alpha helical region, and this is the nexus of both of these filament folds. So there may be some interesting mechanistic link there. You can also see in this sequence alignment that again, the secondary structure motifs do not align well between the brain-derived folds and those generated in vitro. So, we do also need for TDP-43 new models that reproduce these filament folds, and we should be cautious of using in vitro generated assemblies for all of these proteins. This then brings TDP-43 together with tau and alpha-synuclein into this broader notion that we have distinct filament folds that define different diseases. And you can see that there very much for TDP-43 type A and ALS and FTLD type B. Of course, there are more TDP-43 proteinopathies that we are now very interested in and following up. But I'm going to switch now, and for this second part of the talk, focus on another RNA binding protein implicated in neurodegenerative disease, that of FUS. So FUS accounts for the remaining 10% of cases of FTLD, so it was thought, and for a rare proportion, about 1%, I think, of cases of ALS. Now, FUS, as I mentioned, it's an RNA binding protein. It almost has the reverse domain organization to TDP-43 in that it has its low complexity domain at the N terminus. It's a very different low complexity domain to that of TDP-43. It has many tyrosine residues, but again, many glycine residues as well. It has a single RNA recognition motif, but it makes up for that with many arginine-glycine-glycine repeats that are enriched in these two domains here. And then at the extreme C terminus, it has its nuclear localization signal. So the evidence for FUS assembly in neurodegenerative disease came principally first from the discovery of mutations that lead to dominantly inherited forms of ALS. And those references are shown here. Now, I just make the point here that this is only for ALS, and there have not been mutations found in FUS that give rise to FTLD, although many studies have tried to look at this. Now, these studies also showed that FUS antibodies against FUS would stain inclusions in these cases of ALS, and subsequently Manuela Neumann, together with Ian McKenzie, showed that FUS could also stain a subset of cases of FTLD. So that then brought FUS into FTLD as well as ALS. However, a couple of years later, work from the same group then showed that actually in FTLD, you also have two other proteins, TAF-15 and EWS, that are present in these inclusions, and also a fourth protein, transportin-1, which is the nuclear import receptor that binds to the nuclear localization signal of these three proteins, FUS, TAF-15, and EWS. And together, these three proteins are known as the FET proteins. So they have this homologous domain organization that you can see here, and they carry out similar roles in the cell as well. So we were interested about amyloid filaments in FTLD FET with the hypothesis that they would be formed of FUS based on the genetic evidence in AFS. And so we obtained 4 cases, samples from 4 individuals with this disease, and we confirmed together with Bernardino Getty and Tamarine Lashley at Indiana and UCL respectively, that indeed the inclusions in the neocortex of these individuals were positive for FUS and TAF-15 and transportin-1. You can see with these magenta arrows. We didn't find much EWS immunoreactivity. You can see this cyan arrow shows what we saw mostly, which is this nuclear physiological localization of EWS. But EWS immunoreactivity of inclusions was described as being more sparse in any case in the literature. So we then applied this extraction method that we developed for TDP-43 to these amyloid filaments. And we plotted that final insoluble fraction for these 4 proteins. And actually, we found they're all present in this extract. So yes, that could mean that all 4 of them form amyloid filaments, although that would be very unlikely, we think. And actually, what we think is this shows that actually the extraction method is not optimal for these proteins, and that we're getting the physiological form of these proteins coming through in these preps, and they are even more insoluble than TDP-43 in health. And this is really supported from work from that paper I showed in the previous slide, where they did a similar extraction method on neurologically normal individuals, so they didn't have any abnormal assembly, and they still found at least TAF-15, EWS, and FUS coming through in their final fraction number 4. So, this is not very helpful in telling us what is forming abnormal assemblies. These could just be the physiological assemblies of these proteins. So to get at that, we had to really had to use cryo-EM in this instance. So this was a study led by another postdoc in the lab, Stefan Teta, another fantastic postdoc, and he extracted those filaments and looked at them by cryo-EM. I'm just showing you the micrographs here. We can see the ice in the holes in the grids, and then in the corner here, this is the carbon support that we have on these EM grids. And you can see many, many filaments in each field of view here. Now, the yellow ones we'll come back to. You can see they're shared between all individuals. But we also saw some other types of filaments only present in certain individuals. The magenta arrows point to collagen fibers, just so you can get a sense of the scale here. That's one of the contaminants in these preps. And the blue arrows point to these other amyloid filaments. Now, if we focus on individual 4 and on those blue arrowed filaments, Stefan determined their structures and showed that they are TMEM106B and beta amyloid 42. So TMEM106B, we previously contributed to a study from the lab of Michel Goda and Chaussieres, so this reference down here, where we showed that these filaments form in the human brain in an age-dependent manner. And this happens across all neurodegenerative diseases, and it happens in healthy aging as well. And that was a big surprise, and we don't fully understand, what they're doing, and it's likely they're contributing to the risk of developing neurodegenerative disease, but they do not, as of themselves, cause a neurodegenerative disease. Now, we saw them in individual 4, who was older, but we had individual 1 who had died at 30 years of age, which had no TMEM filaments whatsoever, so supporting this age dependency. And then only for individual 4, we had these amyloid beta 42 filaments with one of the structures we previously seen in this collaborative study, the reference shown down here, again with Michel and Shores. And this really matches the neuropathology. We did see some sparse plaque co-pathology in this individual. But returning to the single common filament type that's shared among all individuals, and you can see that's the unique filament type in individual 1. We have no other amyloid filaments. We then focused on this as our candidate hallmark filament of this disease. So Stefan was then able to use cryo-EM again to determine the structures, and he found a single filament structure with an identical fold. And we like to name these folds to give them memorable names, and Stefan immediately thought of a Vespa scooter when he saw this shown here at the scale. And so we named this the scooter fold. I'll refer to it as that from now on. And as I mentioned, we have this identical filament fold shared amongst all individuals. Now, at the resolution of about 2 angstrom for individual 1 or just below, we had a really good map that we could then essentially sequence the protein from this cryo-EM map, so from the structure itself. And probably you can already make out amino acids with large aromatic ring side chains here. You can see many of them. So, okay, this looks like many tyrosine residues. We know that FUS indeed has a low complexity domain enriched in tyrosines. But we really struggled for a long time trying to build in FUS. And Stefan eventually went away and came back and showed me this. And so this unambiguously is the correct amino acid sequence and the correct atomic model for this structure. And just to sort of convince you of that, if we have a look at a bit of a higher threshold, you can actually see the ring, the hole within these aromatic rings. So there are these tyrosine residues, and this sequence then actually turns out to be not FUS, but it's homolog TAF-15. So you can see here this amyloid core. It's this half, the first half of the low complexity domain, the very beginning, the N terminus of this TAF-15 molecule. You can see it's an amyloid filament because, again, it has these beta strands that stack to form intermolecular beta sheets. These areas highlighted in yellow. And then something I didn't mention earlier, but as for all of these amyloid filaments, there are then large flanking regions which are present but not ordered relative to the cores of these filaments that form what we call a fuzzy coat. And these TAF-15 filaments have a huge fuzzy coat incorporating the RNA recognition motifs and the arginine-glycine-glycine motifs. So what we think is that actually in frontotemporal lobe degeneration, these cases that were defined as FTLD FUS should actually be defined as FTLD TAF-15. And we're following up this now, on this with additional individuals with this disease, and also trying to see if we may also have subtypes of FTLD TAF-15 similar to tau, TDP-43, and alpha-synuclein, and if this notion of disease-specific filament folds also holds true for TAF-15. Now, what about FUS? I mean, as I showed you, there is FUS in the sample. There is FUS that, at least using antibodies, can be detected within the inclusions. And I think there's a few possibilities here. Firstly, it may be that the FUS is present in filamentous form at a very low abundance. So we know from carrying out tests that the sensitivity of cryo-EM is about 1%. So if there's a population of filaments below 1% of the total, we probably would miss it. So we can't exclude that. Secondly, it could be that our extraction method doesn't capture FUS filaments. And we should keep this in mind because we didn't see TMEM106B filaments or TDP-43 filaments with the previous extraction methods. And suddenly we did when we tweaked the method. To have a look at this, we checked the soluble fraction from our extraction, and we didn't see any filaments, whereas we could in the insoluble fraction. And we've also subsequently determined structures of these filaments in the absence of pronase, which we use to clean the background in these EM grids. And again, we find an identical TAF-15 structure and no evidence of FUS filaments. So the third possibility, which I favor, is that FUS may be non-filamentous and drawn into these inclusions by TAF-15 filaments. And in that sense, it would be similar to how we think of transportin 1, which we don't think forms filaments, there's no evidence for that, but which we know can bind to the NLS of TAF-15. And in the case of FUS, it could be that FUS is binding to transportin 1 itself. It could be that FUS is binding to shared RNA targets between it and TAF-15, or it could be there are direct interactions between FUS and TAF-15. And there's a basis for all of this in the literature. Again, we would like to test this in experimental systems. Now, just as some sort of way to get at this non-filamentous form, we carried out mass spectrometry on the filament samples. And what we could show is that in cases where we had known amyloid filaments, like TMEM106B or A-beta-42, we could find peptides from the filament cores. Whereas in cases where there were no filaments, we don't have peptides. And that's certainly true for TAF-15 as well. In individuals with those filaments, we find these core peptides, and they're absent in control individuals. But for FUS, there's really no difference in the peptides between individuals with FUS-positive inclusions and individuals without. So I think that also speaks to a non-pathological form of FUS present in these inclusions. Now, what about FUS in ALS? I mentioned that we have mutations causing inherited forms of ALS, mutations in FUS, and actually the inclusions in those cases are not immunoreactive against TAF-15, so it cannot be TAF-15. So it may well be that there are some rare cases of ALS with FUS mutations where you do have FUS filaments, and we're very interested in that. But what we also found in 2 of our individuals was that they actually had symptoms of ALS as well, both clinically and neuropathologically. This is work carried out by Bernardino Getty in Indiana, and you can see here in this Loxel fast blue staining of spinal cord that we do have loss of cortical spinal tracts, which really looks like the neuropathology of ALS. And indeed, we had TAF-15 positive inclusions in motor regions in these individuals, and Stefan was able to solve the structure of amyloid filaments from the motor cortex and brainstem and show that they are TAF-15. So it may be there's an underappreciated new form of ALS that actually has TAF-15 pathology at its core. Now, of course, I started this talk by saying that there are mutations in the genes encoding the proteins that assemble, and that's strong evidence for their causal role. And so what about TAF-15? Well, the studies haven't really been done. There are a few references suggesting pathogenic mutations, but really these are not that convincing. They could just be polymorphisms. The family studies haven't been done, the associated neuropathology hasn't been checked. But I think, if anything, our studies should urge geneticists to do these kind of studies and to look for TAF-15 mutations in ALS and FTLD. So in summary, I have shown you that TDP-43 forms amyloid filaments in ALS and FTLD. So although it's not binding amyloidophilic compounds, it nevertheless is forming amyloid filaments, and the structures give some explanation as to why that is. I've shown you then that TDP-43, tau, and alpha-synuclein then have a general notion in that distinct filament folds define different diseases. This is quite an amazing finding, I think, but one which requires further work to fully understand in terms of its role in the disease process. And most recently, I've shown you that instead of FUS, actually TAF-15 amyloid filaments characterize FTLD FUS and possibly also some cases of ALS as well. And there's been very little work done on TAF-15 mechanistically, and certainly this suggests that we should place a greater focus on this. So I'd like to acknowledge the many people involved in this study. My laboratory here, shown in the top left-hand corner, especially Diana Arseni and Stefan Teta, who led the cryo-EM structures on TDP-43 and TAF-15 respectively. Also to Heidi Chen, who set up the mass spectrometry in the group and carried this out for type A FTLD TDP. Alexei Merzin, who at the LMB, who has really been instrumental since the very first cryo-EM structure of tau filaments in Alzheimer's disease in interpreting these structures, and Sue, who carried out much of the mass spectrometry data collection for these studies. Also thanks to the LMB electron microscopy facility and scientific computing facilities who supported this study. And then in the middle here, to the many collaborators who have provided the human tissue samples that we have used to determine these structures. And of course, the most thanks goes to the patients and their families who have donated these tissue samples, without which we couldn't do any of this work. And then I'd also like to thank here the funding sources, principally the MRC Laboratory of Molecular Biology. So I'll stop there. I'd be very happy to take any questions. Thank you very much.
Thank you. Thank you very much, Ben. So if you have questions for Ben, please type it in the chat. So we have a few questions already. Some of them came early on your talk, so those might have been answered. So one question was, do all amyloid filaments from different patients with the same pathology, would they show the same structure?
Yes, so tau, alpha-synuclein, and beta amyloid, so far we have a correlation between the disease and the filament fold, yes.
Yeah, and also quick technical question, I think, is the 3D structure also determined of the TDP-43 with cryo-EM?
Yes, sorry, yes, maybe I didn't make that clear, but it is. We're using the same cryo-EM and helical reconstruction approach for TDP-43 as well.
Yes, and the other question was about your use of gold. So why did you use the gold? Is it because of the weak electrostatic interactions? What's the reason?
So the gold was only used in the negative stain, so it's separate from the cryo-EM. And this is just used as an electron-dense label, so we can see, we conjugate that to an antibody, and then we can immunolabel, in this case, the filament to identify the protein. And to provide evidence for which protein it is forming the filaments. Okay.
So was AlphaFold be able to predict anything similar?
No, it can't. And yeah, maybe one day. That it could be there just not enough of these structures out there for AlphaFold to use. It could also be that these haven't been under evolutionary selection. Maybe that won't work, but there's something we're interested in.
Okay, and there's the other question about, do you think you can disrupt or prevent filament formation with small molecules?
Yes, that slide I showed for tau, there was some work there from David Eisenberg's group. I think they've sort of been pushing this forward the most, and they've shown there are small molecules that can wedge themselves into even mature filaments and rip them apart essentially. So these people used to talk about amyloid filaments as being irreversible. I think we're seeing now that's definitely not the case. They're very strong interactions, but they're not invincible. and as well as small molecules, the cell has mechanisms, for example, autophagy, the lysosome, which can turn over these filaments if the capacity is there. So, you know, there is hope there for doing this, yes.
Yeah. So what are the factors causing mutations in FUS, causing neurodegenerative diseases? Yeah.
Sorry, could you repeat the question?
Yeah, what are the factors causing mutation in FUS? Do you know why it's been mutated?
Why is it mutated? Well, no. I mean, the mutations, in terms of the effects of the mutations, they cluster mainly within the nuclear localization signal. So it's, you know, probably preventing FUS from getting back into the nucleus, and that would allow its aggregation or predispose it to its assembly because it would remain cytosolic. So that's quite different for FUS, because for the other proteins, the mutations are mainly in the regions that actually form the filaments, and they seem to, you know, in some cases they lead to a more stable filament fold, or in some way increased filament formation. So, you know, FUS is there are many reasons that FUS is quite different to these other proteins.
Have you looked at TDP filaments from any other models, like mouse models, and did you compare it with human?
So this is a large project going on in the lab right now with collaborations. So yes, we're trying to look at cell, mouse, and in vitro models for TDP-43 and to understand if they can recapitulate these structures seen in disease. Right now, we don't know, but we hope to know soon.
And there's another comment on this, a very nice presentation. My question is, can we also use cryo-VR for this purpose?
Cryo-E, so
VR.
Maybe a typo in referring to cryo-ET, electron tomography. In which case, yes. So we are carrying out cryo-electron tomography studies. We're principally using this to look at the molecular environment of these filaments, because the benefit there is we can do in situ studies, and we are trying to solve filament structures from the cryo-ET data using subtomogram averaging. I think that will become a reality quite soon. At the moment, the resolution is not quite there. We don't expect to see huge differences, but we did, you know, recently we had a study looking at extracellular vesicles for tau filaments, where we saw some structural differences, and we saw many molecular interactions within the vesicles that were previously unknown. So I think cryo-ET will be very exciting for neurodegenerative disease research, and will also reveal much more atomic or molecular pathology, if you like, that we can't get with these single particle approach.
Thank you. I'll just take one more question. So, so it's a very nice talk. And have you looked at the electrophysiological response, like EEG, and then comparison between basically the functional and then these aggregates?
Yeah. So yeah, that's a great question, and kind of comes down to the consequence of aggregation, and, you know, how do these actually cause neurodegeneration. So we're doing, you know, others have done this, and we're doing this as well in model systems. And of course, we want to check the structures in these model systems. But it has been shown that TDP-43 assembly leads to toxicity in these kind of models, in mouse models, and in cell models as well. And a lot of this may be focused on the synapse, not just for TDP, but for many of these proteins. But I think that's, you know, that's all quite unknown, and a lot of work to do there. Yeah.
Yeah. So thank you very much, Ben. I think there are a couple of questions, but we can look at them later. So thank you very much, everyone, for attending, and the speakers. So you can continue the conversation online. So you can follow at Biochemical Soc, and also at PPP Publishing on Twitter or X. So, and also we welcome suggestions for future topics and speakers to feature this Biochemistry Focus webinar series. If you have an idea for a webinar in 2024, we invite you to submit a proposal for your upcoming webinar. We can find more information in the Biochemical Society website. So all of our upcoming webinars are listed on our website as well. And if you have missed any of these 80 plus webinars that you've already run, you can watch them again. You can visit the website or YouTube channel. And the recordings from today's webinar will also be available to watch within the next couple of weeks. So the next one, join us on 11th of April at 2 p.m. For your next webinar. So that's the topic on understanding transcription, one molecule at a time. So where we will be joined by Dr. Tinky Lenstra, who will explore her lab's research into understanding mechanisms regulate our transcription dynamics in a single cell. So finally, I want to highlight it is more important than ever to stay connected and engaged with your fellow molecular bioscientists. So join the Biochemical Society community of researchers. So we host lots of different types of courses, meetings. You will have exclusive access to a wide range of grants and bursaries and personal online access to the journals and more. So thank you very much and goodbye everyone.
Thank you. Goodbye.